My name is Max Sherman, Agricultural Sciences and Technology instructor. Our ag program is built around science-based research, where students work with community partners, state and federal agencies to solve issues within the ag and natural resource area. Over the past several years, students have worked on over 25 major research projects within our local community, presenting their results to local, state, and federal agencies. These projects help solve issues within our community. Students also share their projects and present them at local science fairs, the FFA State and National Agri-Science Fair and Intel Science Fair. This last year we also had students competing at Intel's International Science Fair in San Jose, the International Sustainable World Energy, Environment and Engineering Project Olympiad in Houston, Texas, and the Incepo Science Fair in Amsterdam. Through their presentations at local, state, and national and international science fairs, Tillamook's agriculture students have earned nearly $300,000 in scholarships in the past 18 months. Students are also looking at careers in science and bioresource engineering. We have many non-traditional students involved in the program, and young ladies are individuals that are leading much of our research. Two of my students will be sharing with you one of their projects, which is dealing with an invasive species where they have made a startling discovery. And they will share that information with you. Thank you.
Hello, my name is Alice Barline. I'm Kristen Haggerty, and we would like to tell you about a remarkable agri-science discovery about New Zealand mudsnails. For the past 2 years, we've been studying them in the waters in Tillamook County. They are an invasive species that can have up to 700,000 snails per square meter, and they crowd out all the other food for salmon, and they have no natural predator here. Over the past 3 years, we've been doing research to find out kind of where the New Zealand mudsnails are in Tillamook and what kind of salinity levels they're living in. We had traps placed in freshwater, in brackish water, and recently we put them in Tillamook Bay, where it's all salt water all the time. Last spring, in doing research, we discovered the first known population of New Zealand mudsnails to have adapted to live in completely salt water. When we discovered this, we wanted to know what has allowed them to adapt to salt water in a way that none of the other snails have been able to do. So we decided that we were going to do DNA testing and compare the genes of snails living in fresh and brackish water to those living in salt water. So first we had to isolate the DNA of snails from 4 different locations with various salinity levels. After we had the DNA isolated and mixed with 4 different primers, we had to put it into this thermocycler, which does PCR, and it basically creates many, many copies of the DNA.
Once the DNA has been replicated, we take out the DNA and put it in the agar wells, and then place the agar well into the electrophoresis well.
Once the gel is inside of the well here, we're going to cover it with distilled water. Once it's covered in the water, we put the lid on and turn the machine on. What this machine does is push electrical pulses through the gel, separating molecules based on size, and it will create banding in the gel.
So after the electrophoresis process is complete, we take the gel out of it and put it in a tray, and we dye it with a blue dye to reveal the DNA banding. And then we take the gel and we put it under a light to reveal the DNA.
Next, we put this piece of plastic over the top of it, just so that we can outline and we can mark the banding. And once we find a band, we just put a mark there. And then we take the piece of plastic after we're done. We blew up the plastic so that we could see clearly the banding, and we measured
from the wells to the bands of each primer and of the 4 separate gels from the different locations, and we ran ratios to compare the 3 of the 4 primers we used revealed unique base pairings in the snails living in saltwater. This seems to indicate that there's a possibility of a genetic modification in snails living in saltwater. We plan to run replicates of this process and work with Oregon Department of Fish and Wildlife and other agencies to further our research.
So we are very fortunate to have Max Sherman and Claire Thomas working with our young ag researchers and young ag students participating in the research and the search for the species and the problem solving they do with real problems out of our community. The collection of data, the analysis, the statistical work they do, and reviewing that information. They certainly model what kids will be doing as they enter the workforce or research in college. There's certainly great preparation for their life after high school if they intend to go into math and science. Again, they bring relevant real-world problems to the students, and the students tackle those. They do the research that involves accumulating data, collection, research, and synthesis of the information they collect. Certainly a well-balanced program, blending the agricultural sciences with the statistical and math needed to understand the information they collect. So again, we're really fortunate. This is a strong program, and we are blessed to have them here, and they have a terrific connection with kids. We're lucky.